Um,
Okay, go ahead. Thanks.
Okay, thank you. First of all, I would like to thank the International Cancer Conference organizers for inviting me to give a talk in this conference. So, as you can see, I'm attending from Turkey, from Dokuz University, and I work also in the İzmir Biomedicine and Genome Center. The title of my talk is about multiple myeloma. It is SOX7 is a candidate tumor suppressor gene in multiple myeloma. As you may be familiar with multiple myeloma, it's a kind of a cancer of plasma cells. So plasma cells are the cells that generate antibodies, specific antibodies against specific antigens. So they are immune cells. They are very important immune cells. If they proliferate, if they grow in an uncontrolled way, they generate these cancers that we call as multiple myeloma. We work on hematological cancers, and there's a very diverse group of different type of cancers. There are different type of hematological cancers. But one thing important for multiple myeloma is that it is a widespread hematological cancer. As you can see in the slide, it represents 10% of the hematological cancers, and it's characterized by the tumors formed in the bones, in many of the bones. There are a lot of treatment options available, and it's evolving every day, such as proteasome inhibitors, immunomodulatory agents, or autologous stem cell transplantation that extends the survival of the patients. However, it is still an incurable disease, so it means that even if you can temporarily get rid of the cancer, finally relapse happens, and there are sometimes multiple relapses may be observed. So there's an important paper published in Cancer Cell by Laura et al. In 2014 that evaluated the copy number alterations in multiple myeloma tumor samples. First of all, I would like to say that although there are some studies that identified cancer-related genes like oncogenes or tumor suppressor genes that are involved in the development of multiple myeloma identified, but there are still many unknowns about the etiology of multiple myeloma. I mean, the type of molecular alterations. So in this Cancer Cell paper, they identified homozygous deletions of 8p23.1 by using Affymetrix SNP arrays. Why is it important? As you know that recurrently deleted regions in the cancer genomes are enriched in tumor suppressor genes. So it's a good starting point for actually identification, discovery, and characterization of tumor suppressor genes. So in this deleted region, there are 4 candidate tumor suppressor genes: BLK, MSRA, Phoenix1, and SOX7. I bolded SOX7. We chose to study SOX7 because when we look at the literature, although at least there is one paper for each of the other genes that they may be implicated in other type of cancers as a tumor suppressor, but there were more information about SOX7 regarding tumor suppressor role in different type of cancers. This slide is showing actually 3 of these publications that imply that SOX7 is a tumor suppressor gene. There are more than that actually. I just show some major ones. In 2009, Zhang et al. Showed that in colorectal cancer, SOX7 was silenced through DNA methylation. And I will show you that the promoter region of SOX7 is a CPG island, and this may be hypermethylated, so that this will lead to transcriptional silencing. In the colorectal cancer cells, when SOX7 is ectopically expressed, it led to apoptosis-inhibited proliferation. Similar type of observation was made by another group, Bayan and Toll, in 2013 in non-small cell lung cancer. SOX7 expression was again silenced, and ectopic SOX7 expression in lung cancer cells inhibited cell growth and promoted apoptosis. In stomach cancer by Zhu et al. In 2014, they found that SOX7 silencing is resulting in activation of the Wnt beta-catenin pathway. As you know, Wnt beta-catenin pathway is an oncogenic pathway. So there are many studies implying SOX7 as a tumor suppressor gene in different type of cancers. We had an interesting case actually that we studied and we reported the study as a letter to the editor. This case was interesting because this is a multiple myeloma case. It has two consecutive relapses. And during the relapse time, we were able to get the bone marrow cells, sort the tumor cells, which are CD38 positive, 138 positive, 19 negative. And then we did some genetic and epigenetic analysis to analyze whether SOX7 has copy number alteration and transcription silencing or not. And we observed that SOX7 was deleted. And based on the observation that I'm not showing here, it may be actually a allelic deletion. So there was also transcription silencing of SOX7 during the relapse times in the bone marrow tumor cells, suggesting that SOX7 deletion may have a role in development of multiple myeloma, although we cannot conclude that may be also important in the relapse. So based on this preliminary information, we construct our hypothesis and specific aims as follows. So our hypothesis is that loss of expression of SOX7 tumor suppressor contributes to development of multiple myeloma. Our first specific aim is to evaluate whether SOX7 is transcription silenced through genetic or epigenetic mechanisms in multiple myeloma cell lines. And we investigated the SOX7 copy number in multiple myeloma cell lines, examined the incidence and also the frequency of promoter hypermethylation, and demonstrate whether there is underexpression of SOX7 or not. The second specific aim of this study was to investigate whether ectopic expression of SOX7 inhibits multiple myeloma cell growth. So we investigated whether ectopic SOX7 causes negative selection pressure or not, and then demonstrated whether SOX7 leads to cell cycle arrest or not, and examined whether SOX7 promotes apoptosis or not. I don't show a publication, but there's a paper that's showing that SOX7 can lead to G1 cell cycle arrest. I don't remember exactly which type of cancer, but there's at least one paper showing that it may be involved in G1 cell cycle arrest. So here, this slide is showing the copy number analysis. We use quantitative PCR, and 5 mm cell lines are evaluated. As you can see here, two of the cell lines, I9 and KMS18, has monoallelic deletion based on these results, compared to reactive tonsil plasma cells, which are normal control for the actually the normal copy number SOX7. So this we observed that there may be homozygous deletion to 5 cell lines evaluated. As I said, there is a CPG island around the SOX7 transcription start site in the promoter region. You are seeing here the SOX7 exon 1, and the horizontal green bar is showing the CPG island. So we use the same quantitative method. It's a very much time-consuming method, but it was the best one available to do in our research group at that time. So we evaluated SOX7 promoter methylation with bisulfite sequencing, and we checked the CPG methylation status in 239 base pair region, which is shown as blue shaded part here. And we also included negative and positive control. So these are the results. Each of these vertical boxes are representing different CPGs. So there are total 22 CPG dinucleotides evaluated in the SOX7 promoter. And as you can see, negative control, which is peripheral blood cells of a normal individual, generally have white color, which is indicating that there is no methylation. We also use a positive control, which is in vitro methylated and bisulfite converted DNA. As you can see, the positive control shows a very heavy level of methylation. Almost all of the CPGs are fully methylated. Now, if you come to the results in the multiple myeloma cell lines, what we observed is that, especially in KMS18 cell line, which is panel B here, you can see that there is very high level of hypermethylation. And we can see that also in MM1S cell line, U266 cell line, and IM9 cell line, there is hypermethylation of many of the CPGs. Only there is one cell line which is shown in panel D, RPMI 8226. So all these 5 cell lines are multiple myeloma cell lines. So this RPMI 8226 cell line did not have methylation. So we concluded that 80% of multiple myeloma cell lines has SOX7 promoter hypermethylation. Then we evaluated the transcript expression level of SOX7 in multiple myeloma cell lines, and we observed that at least 3 of the multiple myeloma cell lines have low expression levels of SOX7 compared to reactive tonsil cells. So there was also reduction in the transcript expression levels in many of the multiple myeloma cell lines. With also Western blot, but I want to emphasize that this needs to be optimized. So I don't want to tell something very much conclusive. But in this Western blot experiment, we use a positive control SOX7 transfected 293 T cells, and we evaluated the SOX7 expression in multiple myeloma cell lines. As you can see, we have expression of SOX7 protein and gap agent in control. As I said, based on this result, it looks like there is no protein expression in multiple myeloma cell lines, but we will try to optimize it a little bit better because I was expecting that SOX7 protein band will be more actually darker than what we observed. But very likely, even if there is expression of SOX7 protein, it will not be probably at high levels in multiple myeloma cell lines. So then we continued with the functional experiments and we generate a retroviral construct. So we subcloned the SOX7 coding sequence into a retroviral vector called PMIG. So in this construct, as you can see in panel B, SOX7 coding sequence and the enhanced reinforcement protein are separated by areas sequences. So this means that if we do transduction of the multiple myeloma cells with this construct, we can check the GFP positivity as an indicator that transduction worked actually. So it's a kind of a marker for transduction. And this gives an advantage also to do an assay called GFP competition assay. And we perform GFP competition assay using two multiple myeloma cell lines, which has either hypermethylation or deletion mediated silencing of SOX7. So these are not expressing SOX7. We put empty vector or SOX7 containing vector. So empty vector is the control. We performed flow cytometry and gated the percentage of GFP positive cells in regular time intervals. So we track the changes in the GFP positive cells side by side, SOX7 transfused and empty vector transfused cells to evaluate whether there will be some kind of a difference will happen in time in terms of the GFP positivity, in terms of the percentage of GFP positive cells. And for example, if you overexpress the oncogene expectation would be the selection of the GFP positive cells, so it should increase. If it is neutral, there will not be any change. If it's a tumor suppressor, there will be some sort of a decrease, and progressive decrease can be observed. So these are the results. One of the cell lines is 18 cell line, SOX7 non-expressing cell multiple myeloma cell line. The panel A is showing the text flows. A0 is representing 4 days post-transduction. As you can see, we obtained almost 40% transduction efficiency with the empty vector and separate peaks from both empty vector transfusion and SOX7 PMIG transfused cells. And initially, SOX7 was 18%. We checked the positivity of GFP positive cells, and as you can see, there was not the change in time in empty vector transfused cells, so the procedure was not, and retroviral transduction procedure was not affecting anything that may change the GFP positivity as a control. But SOX7 transfused cells, as you can see, in time the GFP positive cell percentage decreased markedly, but in a progressive manner. We did the same experiment in a second cell line, as I said, MM1 cell line, which doesn't also express SOX7. And again, we were able to get a reasonable amount of transduction efficiency, like 24%. With empty vector, it didn't change much after 8 days, but SOX7 transfused cells, initially there was 6, and this is representative for flows, of course, many different experiments. But consistently, what we observed is that progressive decrease in the percentage of GFP positive cells after transduction in the SOX7 transfused group MM1 cell line also. So GFP competition is roughly tells about tumor suppressor effect of this gene, and this then continued with functional experiments. and we did the apoptosis assay. We used the same two cell lines, performed retroviral transduction, anti-vector transduced cells, and SOX7 transduced in a cell line were compared again. We quantified the NXT5 positive cells in GFP positive group, which are representing the transduced populations, and then compared the apoptotic cell ratios. So this is the result with the KMS18 cell line. We cannot conclude anything here regarding apoptosis. We will be repeating this experiment here, although it looks like SOX7 transduced cells have more NXT5 positive GFP positive cells, but as you can see from the standard deviation, it's probably not significant. So we will repeat this experiment to see whether there is any marginal increase in apoptosis in SOX7 transduced group. However, in the second cell line evaluated in MM1S cells, we observed that SOX7 transduced group had a marked increase in NXT5 positivity, as you can see in the FEX plots, representing FEX plots as well as the bar graphics, and this is done 4 days after transduction. So it is at least in one cell line. Based on our analysis, it looks like SOX7 may be inducing apoptosis. We obtained more conclusive results with the cell cycle experiments and used a similar type of approach. Same cell lines, KMS18 and cell lines, anti-vector transduced, SOX7 transduced. We use FEX to evaluate the cell cycle profiles in the GFP positive populations again. So these are the cell cycle analysis results. The panel A is showing anti-vector representative graphic, and panel B is showing the SOX7 transduced cell line, KMS18 cell line. As you can immediately realize that there is an increase in the cells in the G1 phase, with a concomitant decrease in the cells in the S phase, which is very much actually supporting the idea that probably there may be G1 cell cycle arrest is happening upon SOX7 transduction and ectopy expression of SOX7 in the cell line. When we did the same experiment in a different cell line, MM1S cells, we observed a very similar profile. Again, the cells in the G1 phase was increasing in SOX7 transduced group, and there was a decrease in the S phase, as you can see in the SOX7 transduced group. The panel C is actually showing the difference of SOX7 minus the anti-vector, the control group for each of these different cell cycle phases. So, in conclusion, based on our observation, SOX7 is inactivated through deletion and promoter hypermethylation. Of course, this needs to be checked also in the multiple myeloma tumor samples as well. We did these analyses, the ones that I present are based on multiple myeloma cell lines. But based on this information, there is deletion and promoter hypermethylation. And ectopic expression of SOX7 2, SOX7 underexpressing or not expressing almost in cell lines, led to negative selection pressure, G1 cell cycle arrest. This is for sure consistent for 2 cell line. At least in 1 cell line, it promotes apoptosis. And re-expression of SOX7 may be a therapeutic strategy. Actually, there's a HDAC inhibitor, panobinostat, which is approved by the FDA, that potentially can increase the SOX7 expression, and that may be actually kill the multiple myeloma cells. So here is the acknowledgement slide. I would like to thank to our lab members, especially Arda Ceylan, our master student, Şavcı, Aylana, for contribution to this project. I would like to also thank to other collaborators in Dokuz University and other universities. This project was supported by TÜBİTAK, the main funding organization of Turkey. The project number is indicated. I would like to thank to TÜBİTAK and also other institutions, other sponsor institutions that fund our studies as well. With that, I would like to finish my presentation. If you have any questions, I will be happy to answer.